welcome to phyloseminar.org. The current theme is phylogenetic networks, which generalize trees by allowing loops. In this series of 3 talks, we're hearing 3 perspectives on networks. Today, we will hear from Luay Nakhleh, who has been pioneering statistical inference of phylogenetic networks from multi-locus data. In November, we'll hear from Celine Skornavaka on Occam's razor and phylogenetic network reconstruction. And in December, Cecilia Ney will describe challenges in network inference as well as how one can use networks in phylogenetic comparative methods. I want to remind people that the Q&A app is gone now, so you can ask questions through Twitter or IRC as described in the attending sections of the Phyloseminar website. As I mentioned, today's speaker is Luay Nakhleh. Luay is originally from Israel, where he got his BS from the Technion. After this, he did his MS at Texas A&M and then his PhD in UT Austin under Tandy Warnow. From there, he went straight to his faculty position at Rice University in Houston, Texas, where he has risen to be the chair of the department. Welcome, Luay, and thanks for participating, especially after your recent adverse weather events.
Thank you so much, Eric. And I assume you see the slides and everything is fine?
Yep, looks great.
Okay, so I will talk today about phylogenetic networks. And as the title shows, I will briefly start with the notion of displayed trees, with how research in the area of networks started about 20 years ago, where the focus was. And then I will shift to the statistical efforts that we are doing and explain why we went to a statistical approach and the work that we have been doing. So I'm not going to give a lot of summaries from biology papers to justify why we need phylogenetic networks. But I will basically say that phylogenetic networks are very important for modeling evolutionary histories whenever the evolutionary history does not fit on a tree. I'm showing on this slide a few examples of these processes that do not fit on a tree, including recombination, which is one of the processes that led to early work on phylogenetic networks even from the 70s. These phylogenetic networks are called ancestral recombination graphs in the phylogenetics, in the population genetics community. In the phylogenetics community, we like to call them phylogenetic networks. Of course, horizontal gene transfer in bacteria violate the assumption of a tree. Hybridization in eukaryotes, hybrid speciation, where a new species arises through hybridization. Gene flow within subpopulations, all these processes violate the assumption that the evolution of a set of genomes is tree-like. And this is why we are interested in phylogenetic networks. In computer science jargon or mathematical jargon, a phylogenetic network, the topology of a phylogenetic network, is a rooted, directed acyclic graph. And the leaves are labeled by the set of taxa of interest to us. So here what I'm showing is a phylogenetic network that models the evolution of genomes from 3 species, for example, ABC, from their most recent common ancestor. If you look at, there's a special node in this network, which is I'm representing or showing by a star. This is what we call reticulation node. Reticulation node has 2 parents, and these 2 parents are there to indicate that the genetic material in B could have one of 2 possible evolutionary trajectories. Some regions in the genome of B could trace its evolution back to the most recent common ancestor with A, and some other genetic material in B came through horizontal transfer from C. This is an example of one phylogenetic network on only 3 species, but you can imagine this can have as many species as we want, so in leaves, for example, and we can have as many of these. So, as I said, the early work on phylogenetic networks in the phylogenetics community started based on some fundamental observations about networks and their what we call displayed trees. So what are displayed trees of a network? Let's consider this network here that I'm showing. And B is a hybrid here. Let's call it a hybrid. So again, as I said, some of the genetic material in B, if you go backward in time from the leaves towards the root, some of the genetic material are going to track the black arrow, then the blue arrow. Other genetic material could track the black arrow, then take the green route through that network. That's, for example, for introgressed material or material that were horizontally transferred. Now, let's think about this network and think about a gene, how a gene could have evolved down this network. One possibility for the gene not to use the green arrow, so to speak. So a gene was not horizontally transferred. its evolutionary history would be represented by this tree, T1. But there are other genes that could have been horizontally transferred and not inherited through the blue edge. Their tree would be this one. So these two trees, T1 and T2, these are the set of what we call displayed trees. And the community always thought of a network as being the sum of these trees, or that a network could be characterized by the set of these trees inside it, or displayed in it, or contained in it, all sorts of terms. But these are the two trees that would capture the evolution of every gene inside the network. This notion, or this observation, gave rise to a very interesting computational problem for inference. The input, imagine that the biologist has inferred a set T of gene trees. Then the output, what we are interested in under this formulation, or under this observation of display trees, is a network with the fewest reticulation nodes that displays all of the trees in T. Why the fewest reticulation nodes? In the absence of any other constraints or any other biological knowledge, we go with the simplest possible explanation, and that in this case would be the fewest reticulation nodes. This problem actually became very attractive to the computational and mathematical communities. This model that a network is basically the set of display trees led to an explosion in development of complexity results, exact algorithms, mainly fixed parameter tractable, approximation algorithms, heuristics for inferring it, and lots of special cases of networks were introduced, like GALD networks, level K networks, and so on. These classes were not introduced for biological realism, but they were introduced mainly to deal with the computational complexity of the problem. So many of the questions that we address on these networks become easier from a computational complexity perspective if we assume, for example, a GALD structure of the network. GALD structure of the network, roughly speaking, means that the hybridization events or reticulation events in the network are independent. They have nothing to do with each other. I will not be talking much more about this formulation, which I call a parsimony formulation, because as Eric mentioned, the next Philo seminar in this series about phylogenetic networks by Celine Skornavaca on November 22nd will focus on this aspect of networks. And I will leave the space for her to talk about the results, and she has contributed significant results in that area. So I will now start moving towards the statistical inference, but before I do that, let me just illustrate to you how this parsimony formulation would be used in data analysis. So a biologist interested in inferring a phylogenetic network would do the following. They would obtain the sequence data first thing. So for example, here the biologist has a set of 5 species or 5 individuals from these species. They go and sequence a set of loci of interest, in this case 6 loci. Let's assume they are independent, unlinked in the genome. The next thing they would do is they would infer a gene tree for each one of these loci. So they end up with a set of 6 gene trees. How you build a gene tree? You could do neighbor joining, you could do RexML, you could do Mr. Base, it does not matter. And now that we have the set of gene trees, we go to the formulation of the problem and we say, give us the network with the fewest number of reticulations that would display this set of trees in step 2. Okay. Now, this model has limitations in practice. The first one is that the smallest number of reticulations could be arbitrarily far from the true number of reticulations. What I mean by this is that in reality, in practice, the set of species you are looking at could have hybridized 10 times. However, a parsimony formulation can give you a network that has 1 reticulation in it. For example, one simple example to illustrate that, if the hybridization happened between 2 sister species, from a parsimony perspective where you focus on the topology of gene trees in the input, that's not something detectable. The other issue is that even if the minimum number equals the true number, suppose that there were really 3 hybridization events, and the solution to that parsimony problem tells us that the minimum number is 3, the set of 3 reticulations that we could find from this parsimony formulation is not necessarily the true one. Of course, one can think of, let us compute the set of all possible parsimony solutions, but that makes the problem even more challenging. A third thing is that the model does not allow for estimating parameters of interest to biologists. So the formulation I just showed basically gives you the topology of the network, and from that you can get the number of reticulations. However, it does not allow us to estimate branch lengths, does not allow us to estimate anything about mutation rates, or about probabilities or migration rates, and so on. And the fourth one, which is really the main motivation for me to justify why we need to go to a statistical approach, is that processes other than reticulation could give rise to gene tree incongruence. What I mean by this is that when you look at a set of gene trees in the input, and they are different, there is some incongruence among them, that does not necessarily mean every instance of that incongruence is due to hybridization. For example, here's an illustration from a very good paper that appeared in the literature a couple of years ago in the journal Science. And in this work here, the authors wanted to study the evolutionary history of Anopheles gambiae mosquitoes. What they did is that they took the genomes of these 6 species that they are denoted here. If you see in this panel, they are denoted GCAQLR. These are 6 different Anopheles gambiae genomes. What they did is that they aligned the genomes, then they passed a window across this alignment, and within each window they built a tree. At the end of that exercise, they tabulated the trees to see every tree, how often does it appear. For example, do all the windows give rise to the same gene tree, or do we see more gene trees? What this panel is showing you is the extent of heterogeneity or incongruence across the genomes of these 6 species. So what you are seeing on the right-hand side of that panel are 9 possible gene trees, and these were not the only ones. These are the major ones, the ones that appeared with high frequency. But what this shows is that, you know, the evolutionary history of these 6 species, as reflected by the evolution of their genomes, is very complex. Different regions are evolving down different gene trees. And the question that the authors were interested in is that what is the evolutionary history of the species themselves? And this is the evolutionary history that they reported in the journal. And the black lines here denote what they refer to as the species tree. So they still delineated a structure that they called species tree. It was mainly based on the X chromosome data. And you are seeing there 3 green arrows on top of that tree. These were postulated hybridization events that the authors said they are needed to explain the data. I would say that these hybridization events were inferred based on to a certain extent, manual analysis of the data and inspecting divergence times that they estimated based on the gene trees and so on. In the same issue of the journal, a nice perspective was written by Andrew Clark and Philip Messer, in which the authors said that given the extent of hybridization that they are seeing, that the authors are seeing, and the extent of incongruence, maybe we need to dispense with the tree and acknowledge that these genomes are best described by a network. And this is in contrast, or does not agree with what the authors did, because the authors, as I said, first inferred the species tree and then they inferred horizontal events on top of that. The message in this perspective says maybe we should not worry too much about an underlying species tree and horizontal edges on top of it. In addition, what Clark and Messer said is that there are 2 possible reasons of why these gene trees are incongruent in this study. One of which is introgression, which is a form of gene flow or horizontal transfer. And the other one is incomplete lineage sorting. So the issue that was at hand here is that we really cannot look at a set of gene trees in the input and say all incongruence is due to hybridization, let's infer a network. Because we could easily end up overestimating the amount of reticulation because we did not account carefully for other processes for that incongruence that had nothing to do with hybridization. Our group, me and my former student and research scientist, we in fact took this notion of displayed trees, and we still stuck with the parsimony approach, and we published this paper in Systematic Biology in 2013. In this paper, we extended a criterion that Wayne Madison had proposed in 1997 known as minimizing deep coalescences. Wayne Madison proposed it for species trees, and he said, let's infer a species tree that minimizes this combinatorial criterion. Here in this work, we extended it to networks to account for incomplete lineage sorting and reticulation as well. I am not going to go through this work because the work has its limitations. The first one is that this is a parsimony approach. It suffers from the typical problem of parsimony. MDC, minimizing deep coalescences, has been shown to be not statistically consistent, even in the case of trees. So it could give you the wrong tree, even with large amounts of data. The second thing, it does not allow us for inference of parameter values. It did not allow us to infer branch lengths, mutation rates, and so on. And it was not very clear for us how to obtain the correct number of reticulations. We basically had to add reticulations one at a time, and then inspect the networks and their scores to hopefully visually be able to tell what was the number of reticulations. And this is why we moved towards a statistical inference in the hope that all these questions or all these issues will be dealt with in a principled manner. So before I go into networks from a statistical perspective, I have first to talk about trees, because really our work is not in a vacuum. We built on the great work that was done in the area of trees, especially under the model of multispecies coalescent. So the field of phylogenetics from the era of a single locus moved from this picture here, where we really have, the biologist had the sequence from one locus of interest, and they built a gene tree. The tree they got, it was a gene tree. If they called it a species tree, that's just a decision. But the tree they got was a tree for this locus. And this field, of course, with the ability to sequence more genomes, we moved from this concept of a gene tree to species tree. In this area here, of course, Felsenstein's pruning algorithm was central in allowing us to compute the likelihood of a gene tree efficiently, because this basically resulted in the practical software tools, whether based on maximum likelihood or based on Bayesian inference, for allowing us to infer these gene trees in practice. But with the arrival of whole genome sequencing technologies, now the biologist had whole genome data, or in many cases, data from multiple loci across the genome. And now the interesting thing for them was how to get the species tree from the data that comes from individual loci whose evolutionary trees are not necessarily in agreement, right? So the question was, how do we go from this sequence data to the species tree when we know that this sequence data did not evolve down that species tree necessarily? And the trick to answer that question was the observation that, yes, these loci do not evolve down the species tree itself, but they evolve down a set of gene trees. So every locus here comes with its own gene tree that it evolved under, and these gene trees evolved inside that species tree. So now we can look at this hierarchical process where a species tree gives rise to gene trees give rise to the sequence data. And this trick or this observation is what allows that link from sequences to the phylogenetic tree or the species tree. And this formulation here accounts for this hierarchical process. What this is showing here is that the probability of observing the sequences is, given the phylogenetic tree on top, or the species tree, has to do with the probability of the sequences evolving down the gene trees and the probability of the gene trees evolving inside the species tree. The integration here over all possible gene trees with their branch lengths is basically to denote that this is not something, for example, we are interested in, and we want to integrate over all of that to account for uncertainty, or again, because it might not be the quantity of interest to us. Now, one of the nice assumptions that we usually make, and most software tools make, is that if the loci are independent, then now we can do the calculation for every locus individually, and we can take the product over all loci. Now, the question that, as I said, the probability of sequences given a gene tree, that's computable using Felsenstein's algorithm. But the question that the community faced in this area was what is the probability of a gene tree given a species tree? How do we compute that? And this is where the multi-species coalescent was introduced. And multi-species coalescent is a way of viewing species trees as a set of populations that are glued together by a tree structure. So if we look at this species tree of human, chimp, gorilla here, this is really a tree of 5 populations. There is the human population, there is the chimp population, there is the gorilla population, there is the ancestral population of human and chimps, and there is the ancestral population of all of them. So one could think of each one of these populations as a structure on which to do all sorts of population genetic computations. But where the multi-species coalescent comes, it says we can glue these populations together using this tree structure. And now, with this new structure, we can look at a gene of interest that we sequenced from an individual in humans, an individual in chimp, and an individual in gorilla. And we can start looking at its evolution backward in time. And when we do that, the interesting thing that happens in this illustration I'm showing is only when the ancestral allele of that individual from human and chimp enters the ancestral population. It's at that point where these two alleles have the chance to coalesce together or the chance to keep missing each other, so to speak. In this illustration I'm showing here, what happens is that these two alleles from human and chimp, they miss each other generation after generation after generation until they enter into the same ancestral population with the allele from the gorilla population. And at that point, just by chance, it happened that the allele from chimp and the allele from gorilla coalesced together before either of them coalesced with humans, giving rise to this gene tree that disagrees with the species tree. Viewing this process as in terms of this structure that glues the populations together, the probability of this happening, the probability of observing such a gene tree really has to do with two quantities in this tree that I'm showing. The first of them is the difference between T2 and T1. This is the number of generations that have separated the common ancestor of human and chimp, or the time at which they split, from the time where their ancestors split from gorilla. So this is the number of generations that separate the two common ancestors there. And the other event is the width of a branch here, which is the number of circles within a branch, which corresponds to the population size. And the multi-species coalescent actually allowed for exact computation of the probability of each one of these possible gene trees. What I'm showing you here are the 3 possible gene trees: human chimp versus gorilla, human gorilla versus chimp, chimp gorilla versus human, and the 3 probabilities of these 3 trees as a function of that branch, the length of the branch that separates human chimp from gorilla, and the population size on that. This quantity here, T2 minus T1 normalized by n, or sometimes 2n, this is the length of a branch in what we call coalescent units. The panel on the right here is visualizing these 3 probabilities, and the main point that it is showing is that as the length in coalescent units of that branch increases, the dominant tree is the one that agrees with the species tree. So the most likely gene tree is really the one that agrees with the species tree. This is a result that holds only for species trees with 3 taxa. Beyond that, for 4 and 5, especially 5 and more taxa, that's not always the case, that the gene tree with the highest probability agrees with the species tree. There is a very seminal paper on that by James Dignan and Noah Rosenberg, where they talk about the notion of anomalous gene trees. This, based on the multi-species coalescent, there is the seminal paper by Ranella and Yang in Genetics 2003, where they derived the density function of gene trees under this model. And then 2 years later, James Dignan and Laura Kobatko today, used to be Salter at the time, they give the probability mass function of gene trees given a species tree. And these 2 calculations gave rise to many methods for inference of species trees from multi-locus data. Again, I'm interested in, if we go back to the story of the mosquito genomes, how do we now account for incomplete lineage sorting and reticulation, as Messer and Clark said? So our work really builds on the work that was done also by Laura Kobatko and her student Chen Ming at the time, where they basically said that you can look at a phylogenetic network, the one on the left here, where B is a hybrid between A and C. You can look at it in terms of the two displayed trees, and they are on top and at the bottom there. And you can think of them a weighted sum of these two trees, or a mixture model of these two trees with weights gamma and 1 minus gamma. I just showed you the probabilities of gene trees under the multi-species coalescent. Now, what you are seeing on the right here are the distributions of gene trees for each one of these two. And now you can think about weighting these probabilities or this distribution by gamma and 1 minus gamma. And this is what they did. And this is the likelihood function here. Of course, in this likelihood, they fixed the topology of the network. It was this specific 3-taxon topology. So the parameters of interest are the gamma and the branch length t. And this likelihood can be formulated in terms of the distributions on the gene trees and gamma. This formulation by Laura Kobatko and her student depends on the notion of display trees, but unfortunately, display trees work only in such simple scenarios. Let me illustrate. Look at this scenario here. We went beyond the 3-taxon. We have now hybridization event followed by speciation, for example, or divergence of B and C. Now look at the red gene tree inside that network. So that red gene tree from B and C, when going backward in time, it failed to coalesce before it hits the reticulation node. The allele from B went left, the allele from C went right. This scenario cannot be captured by either of the two display trees inside that network. Instead, we really need now to look at the network in terms of a set of what we call parental trees. So the set of trees at the bottom, these are the 4 parental trees inside that network. And in the presence of incomplete lineage sorting, these 4 trees are needed. They are more than or subsume the set of display trees if you want to capture the evolution of a gene tree. So the notion of display trees, as we saw it before, is not sufficient to capture the evolution of gene trees in the presence of incomplete lineage sorting. Okay? And my students and I wrote a paper about this last year where we talked about this notion about what is a tree inside a network and so on. So this is what led us to the multi-species network coalescent, where we said let us try to extend the multi-species coalescent, where the populations are not linked by a tree structure, but they are linked by a network structure. So what we did here is we went to the same thing as we saw before, phylogenetic networks, but now we parameterize these phylogenetic networks. In the parsimony formulation, we did not care about branch lengths of these networks, but now these branch lengths, these networks have branch lengths with them as well in coalescent units, analogous to species trees. And in addition to that, just as in the work of Laura Kobatko and her student, also we have this gamma here, inheritance probabilities. I am showing here a little bit more complicated situation of inheritance probabilities to denote one issue that could happen in practice, where different loci in different individuals in the species of interest could have their own inheritance probabilities, could have their own gammas. So here, for example, in this figure, what I'm illustrating is, imagine the biologist had really sequenced 6 individuals from B. So every row of these 4 shapes here is one individual from species B. And every column in these 4 rows, in these 6 rows here, is an individual locus. What you look at here, for example, the triangle locus, it has the signal for that is all coming from the side of A, whereas the signal of the diamond, it all came from the C direction. What this means is that the introgressed locus that's denoted by the diamond here got fixed in that population, in the B population. However, the other loci, for example, the one denoted by square or circle, they could have different introgression pattern or different frequency in the population of B. This is what I'm trying to illustrate in this figure, that things could get complicated, and one could think of introducing one inheritance probability per locus. Of course, in practice, introducing one parameter per locus is problematic from inference perspective, because as the data increases, so does the number of parameters. And from a mathematical perspective, introducing more parameters does not affect anything. The math would hold. From a practical perspective of inference and from an implementation perspective, we usually assume one gamma for each one of these reticulation nodes. So the gammas, the inheritance probabilities, differ among reticulation nodes. Once we have the topology, the branch links, and these gammas, now we can view the multi-species coalescent, but inside these networks. So what I'm showing on top here is a phylogenetic network psi. And to the right of it, I'm showing a topology of one gene tree. In this case, we have one individual sampled from A, one individual sampled from C, two individuals sampled from B, B1 and B2. At the bottom here, what I'm showing with these 10 scenarios, each one of these is what we call coalescent history. This is, each one of them is one way in which that gene tree could have evolved down the network. Now, this is the notion that Laura Kobatko and James Degnan introduced in their 2005 papers, that you could compute the probability mass function of gene tree topologies on species trees in terms of these coalescent histories. We took that notion and we extended it to networks, and we showed actually how you can get the probability distribution of gene tree topologies within a network by using this notion of coalescent histories. But now they are working inside a network rather than inside a species tree. Later, also analogous to the work of Ranella and Yang, we developed the density function for gene trees with their branch lengths here. So now this accounts for the branch lengths of the gene trees in addition to their topologies. The likelihood calculation, or the calculation of a probability of a gene tree topology inside the phylogenetic network, is the major bottleneck in terms of computational complexity for statistical inference of networks. Because the number of paths that a gene tree can take inside the network explodes exponentially with the number of reticulations in that network. So two pieces of work we did here a few years ago. One of them is that we introduced faster exact algorithms and also faster heuristics for computing the likelihood of a phylogenetic network in terms of the gene tree topologies. In addition, analogous to the work of Liang Liu, Scott Edwards, and others, we introduced the equivalent of MPS for species tree, which is maximum pseudo likelihood approaches for species trees, but we introduced it for phylogenetic networks. I'm not going to go into the details of these, but these were techniques to speed up likelihood calculations to allow us to hopefully scale for larger data sets. So now that we have derived the probability mass function and density function, this allowed us now to do inference of evolutionary histories of species under this, what we call the multi-species network coalescent. So in terms of inference, the data, again, here we assume M independent loci. For each locus, we assume that we have a sequence alignments for that. And we are interested in the phylogenetic network and inheritance probabilities. When I write phylogenetic network psi here, by psi, I denote the topology and the branch lengths of that. The important assumption here is that there are no recombination within a locus, so that every locus has evolved down a gene tree. And there is free recombination between loci, which allows us now to take product over probabilities of gene trees. So this is important to emphasize here, that when I talk about gene trees, these are not protein coding genes. These could be any region across the genome, coding, non-coding, regulatory region, or whatever, exons, introns, as long as they are independent, the loci that are collected are independent, and no recombination within a locus, things should hold here. Now, one popular technique that's used in inference in practice is what I call the gene tree shortcut. Here, assume that the user has built gene trees on each of the M loci. Okay, so G1 is a gene tree on locus 1, G2 is a gene tree on locus 2, and so on. Then what we are interested in is a phylogenetic network and inheritance probabilities from these gene trees. So now the data is really the gene tree estimates. The sequences are no longer relevant. Okay. This speeds up the calculations because we don't have to account for the sequence data and for the gene trees that they evolved under. In this paper here, 2 years ago, we introduced a maximum likelihood approach for finding such a network. So here we saw the network psi, its branch lengths, and these inheritance probabilities that would maximize the likelihood function. And of course, this is a very hard problem. This is a hard problem for trees. It's even worse for networks. So we were not expecting to find an exact solution for finding these networks without basically doing some search of the network space. And that's what I'm showing here in this cartoon, is that we devised a set of operations that will allow us to walk through the network space. These operations, for example, would allow us to add a reticulation or to remove a reticulation or to change the branch length of a network and so on. We partitioned the space of networks in terms of the subspaces based on the number of reticulations in the network. So the lowest layer is the space of all trees. The second layer is the space of all networks with one reticulation and so on. Now, one interesting thing that doesn't happen with trees and happens with network is that now the space is infinite. You can keep partitioning branches as much as you want and to add as many reticulations as you want. So it was important for us to partition like this so that we could search a space, for example, with I-1 reticulations before we jump into the I reticulations, because a very simple result that we all know about is that the more reticulations you add, you are adding more parameters to the model. You can fit the data better, or at least as well as with fewer parameters. And we wanted to try to make sure that we are adding true reticulations and we are not just overfitting data here. I would not claim that this work, of course, addressed that in any satisfactory way, but we introduced some information criteria here as a first attempt to try to account for that issue of model complexity. We used this maximum likelihood technique and we reanalyzed the mosquito data set that was analyzed in that science paper I showed at the beginning. We did this in collaboration with Matt Hahn, who was also an author on the original science paper. I'm not going to spend much time here, but I will just show some of the analyses that this new framework allows. What you are seeing here on the left in panel A is that what we did here, we took the topology that was reported in the science paper. So we took their species tree and their arrows, the horizontal edges, and nothing else. Then we said, let's take the data and optimize the parameters of this network. So what we say here is that if we take the topology of their network and nothing else, and we try to optimize branch lengths and inheritance probabilities, what do we get? What you're seeing at the bottom here is the log likelihood of that. What you are seeing on these horizontal edges, these are the estimated inheritance probabilities. I'm not showing the branch lengths. What you are seeing on the right here is that we said, let's take only their species tree, not their network, but just the species tree, and now try to infer the horizontal edges and the parameters. One of the interesting things that happened here, for example, is that you notice that once we infer the horizontal edges, we found that one of the horizontal events was in the reverse direction from the one they reported in their science paper. So this illustrates the type of analysis you can do. You can fix a network and say, what's the likelihood of that? You can fix a species tree and say, give me the horizontal edges that will optimize the likelihood up to a fixed number of reticulation. Of course, it allows us also to do things from scratch. What I'm showing you in this slide, here we took the data with no network at all, no topology of a species tree, and we said, infer the best species tree from this data. That's panel A. Infer the best network. What I mean by best here is the maximum likelihood. Network with one reticulation, that's B. Infer the best with two reticulations, that's C. Infer the best with three reticulations, that's D. The fourth one here shown in orange, that's from the X chromosome data. So we analyze the autosome data separate from the X chromosome data. What this is illustrating here is that when we did the search from scratch, we got also topologies that disagreed with the original studies here. The major disagreement, I would put it as, it has to do with science paper insisting on an underlying species tree, and then postulating horizontal edges on top of that. In this work, the search procedure does not designate anything as species tree. It searches the network space, and that made a difference in the type of results that we obtained. Then after that, we went towards a Bayesian approach, and the reason we wanted to go for a Bayesian approach because we really wanted to estimate the posterior of networks so that we get an estimate of uncertainty in these networks. We also wanted to, hopefully in a principled manner, account for model complexity through the prior. And we wanted also to try to specify knowledge from the biology of hybridization, for example. So we know that as the species that are closely related, they have higher chance of hybridizing than species that are very far apart. This kind of information can be incorporated in terms of a prior on the network that we search or we try to infer. And this is what I'm showing here. This is a paper we published a few years ago also in PLOS Genetics, where the input here was a set of gene trees, and what we estimated or sampled is a posterior on networks. The results were similar to the likelihood. One major difference here is that we did not need to specify the number of reticulations a priori. We in fact used through the prior, and a prior that penalizes model complexity was sufficient to detect the number of reticulations. And very quickly, I'm going to go through more recent results here. So this is a paper that was just accepted for systematic biology and it will appear very soon. Now we said, let's go all the way from the sequence data towards phylogenetic networks. So let's not use gene tree estimates as input. The reason for that is that first, it allows us to estimate more parameters. In particular, using sequence data allows us to estimate population mutation rate. And the second thing is that when you use gene tree estimates as input, these gene tree estimates are going to have errors in them. And if we go from the sequence data directly, we hope to account for that uncertainty in gene trees in a principled manner, because we are not going to be fixing them. We are going to be sampling or hopefully integrating them out. So now the data is a set of sequence alignments, the original data set of sequence alignments, no gene trees are inferred here. And we are interested in the posterior distribution on the network and the inheritance probability. This posterior distribution is proportional to the likelihood of the network and to prior on the network and inheritance probability. This integration over all gene trees is, of course, computationally infeasible when the data is sequence alignments for the loci. And we try to co-sample the network and the gene genealogies here. Okay. And the prior that we did, I mean, this is an area that those interested in this work here, this is one open question, is to derive principled priors on phylogenetic networks, analogous to birth-death models for species trees and beta splitting models and so on. Here in this work, we did the composite prior where we have an exponential distribution on divergence times of the nodes in the network. We have a gamma distribution on population mutation rate here. So every branch has its own mutation rate, 4 times n times mu. We also have a beta distribution on the inheritance probabilities. And we have a weighted Poisson distribution on the number of reticulation in the network. So this is a Poisson prior normalized by Tn, m, which is the number of networks with n leaves and m reticulations. And let me be very clear about this. We don't know how to compute this number. We get an estimate of it, especially that in the Metropolis Hastings algorithm, we really need to take the ratio of nm and nm plus 1 that allowed us to do some sort of approximations for that quantity. And we also have this exponential distribution on what we call the reticulation diameter. So if you think about this reticulation node, and you can think about the diameter as the length of the path that connect, the sum of the length of the two paths that connect this reticulation to its ancestor, which is the root of the network. We have an exponential distribution on this. And this is what I was saying about incorporating biological knowledge here. What this prior does is that it says, I'm going to give higher probability to hybridizations that happen between more closely related species than farther ones. We had to revise the PDF of gene trees here so that we factor in the population mutation rate rather than just using branch lengths in terms of coalescent units. And we then devised a reversible jump MCMC algorithm here. The reason for reversible jump is because the space we are sampling and walking through is going to change dimensions. The network we can add reticulations, we can remove reticulations. This is going to result in differing numbers of dimensions as we are walking through that space. And reversible jump MCMC is one technique to deal with such spaces. And we developed a long list of operations that allow us to traverse that space along with their Hastings ratios. We implemented all that, and I'm showing here only simulation results. So we, for example, in this case, we took this network as a true network with its divergence times here that are shown at the nodes in expected number of mutations. And there's that variable S at every node. So that's a scaling factor. We multiply each one of these divergence times also by value of that scaling factor to make the instance of the problem either harder or easier. So S equal 0.1, this makes the branch is extremely short, and this becomes a very hard instance. When it is 1, this is a typical instance when, for example, the mosquito data set. Then we generated data on this with varying sequence length and varying numbers of loci. And then we looked at the performance of a method. What I'm showing you here is how the method performs, for example, in terms of estimating divergence time at the root of this network. And each one of these panels corresponds to a different number of sites in the sequence alignment and a different scaling factor. This is the factor that shrinks and expands the divergence times. The lower right panel, the bottom right panel, should be the easiest case for the inference because it's the longest sequences and the highest value of the scaling factor. The top left panel should be the worst case because very short sequences, very short time elapsed between divergence times. Within each panel, the dotted line is the true value. And as you can see, the method does very well in estimating divergence times, almost regardless of the number of loci that are used and almost regardless of the sequence length. Of course, the left column has the highest amount of standard deviation. As we go to the right, the estimates become much more concentrated around the true value. And this is an estimation how the method does in estimating the population mutation rate. And again, remember, parsimony approaches and so on, they cannot estimate these parameters. And similar situation here, the dotted line is the true value. The method does very well in terms of estimating it. The more loci you give it, the longer the sequences, of course, the performance improves. But interestingly, even when we multiplied all these short divergence times by a scaling factor of 0.1, still the method does very well. This is the number of reticulations that we estimate. The red bars here, what you are seeing for every number of loci is 3 bars. The red one or orange, whatever this color is, it is the number of instances where we sampled the tree. The green is when we sampled a network with 1 reticulation. And the blue, which is hardly seen here except for the bottom layer, is where the sampler sampled networks with 2 reticulations. As you can see here in the top panel, for example, the top row, when the branch lengths are extremely short, the method is going to estimate species trees. And this is important to highlight here for 2 reasons. First is that there are these regions where estimating hybridization is going to be very hard because the divergence is so short that the signal for hybridization is not there for inferring them. The second thing that I want to highlight here is that these are not methods that will infer networks no matter what. In this case, for example, in the top row, the method said there is no reason to invoke reticulations. It did not, and it gave a tree back. So this issue is that network methods can also infer trees. It's important to keep in mind. When you run a network inference methods, it's not going to force a network on your data. Of course, when we go to the bottom there, for example, when the scaling factor is 1.0, and this is a typical instance, it's not a very hard instance, you can start seeing now the green bars rising, and that's where we are basically inferring the true network here, or the true number of reticulations. This panel here, this figure is showing whether we get the right number of reticulations, not necessarily the correct reticulation events themselves. The next one is about that. The blue dots here are the distance of the topological distance of the network we infer to the true one. This goes hand in hand with the previous figure, is that if you look at the top row, the blue dots are all high. We didn't get the network at all, and the distance from the true network is high. At the very bottom there, you see that the network, actually the distance goes down to 0, which is the dotted line. The method is inferring the true topology as well. What we are showing in green here is the distance to the true backbone tree. What is the backbone tree? If you look at the network on the left and remove the arrow from there, there is that underlying tree. I don't want to call it species tree. I'm going to call it a backbone tree for computational purposes and to avoid talking about the notion of a species tree. What you are seeing here is that even when we don't get the network, which are the blue dots are high, the green triangles are low and are getting close to 0 even in the second row from the top. What this is saying is that when the method did not infer the network, it inferred the underlying backbone tree, which is the signal for the vertical inheritance here. So the method does very well. And the last thing I want to say is that we also have this paper that's currently, a revision of the paper is under review now in PLOS Computational Biology, where this is a work that built on the work of David Bryant and his colleagues for the SNAP method for inferring species trees from biallelic markers like SNPs and AFLP data. In this work here, the major computational contribution is that we can, similar to the SNAP method where they did this integration over gene trees analytically, but in their case, the species phylogeny was a tree. Here we do this integration analytically, where the species phylogeny is a network. The running time complexity of that is linear in the number of species, but it is exponential in the number of reticulations. Here, so K is the number of reticulations, N is the number of individuals, because you can have, for example, 3 species, but you can sample multiple individuals from each, and that plays an important role in the running time of the algorithm. Similar to before, we use this reversible jump MCMC to sample the posterior of networks, but now based on this integrated likelihood where we analytically integrate the gene trees. It's important to highlight here that this analytical integration works only when we have biallelic markers. So every marker is really one site. We cannot adapt this technique to the sequence alignment, or at least we don't know how to do it. All the methods I described here are implemented in a software package that we have been developing for 10 years now in Java. It's called Phylonet. It's publicly available in open source and executable at this link here. We use some extended Nexus input-output format for it. The networks are produced in what we call rich Newick format, which can be visualized using Dendroscope that was written by Daniel Hewson. Here's an example from the website of Phylonet that shows you the different commands that only just, for example, for inference of trees. So the top one, MCMC-seq, this is the one that does co-estimation of network and gene trees from sequence data, followed by the one that does estimation from the biallelic markers, then the Bayesian method on gene trees as input, the pseudo-likelihood one, then the likelihood with cross-validation and bootstrap to try to account for model complexity. Then we have infer network underscore ML. This infers it under maximum likelihood. And the last one there, infer network underscore MP. This is the one that infers network under maximum parsimony, or the minimizing deep coalescence criterion. To summarize, this multispecies network coalescent that we basically built on multispecies coalescent accounts for both incomplete lineage sorting and reticulation simultaneously. So it allows us to avoid using the parsimony formulation that would treat everything as either incomplete lineage sorting and give you a tree, or everything as reticulation and give you a very complex network. We have developed maximum parsimony, maximum likelihood, and Bayesian inference methods under the multispecies network coalescent. The Bayesian one allows for use of sequence alignment and biallelic markers as the data. All 3 of them allow the use of gene tree estimates as the data. All models and methods allow for multiple individuals per species. So you can have 0 individuals, for example, for one locus for certain species, you can have 5 individuals and so on. Network-based methods can infer trees, as I showed. Tree-based methods cannot infer networks. So when you are unsure, you can just run a network-based methods because it will give you the right thing, in my opinion. The major bottleneck here is the computational requirement. The field of species tree inference has made great strides towards large-scale analysis. For example, the method Astral allows you to infer species trees on hundreds of taxa. We are nowhere near that in terms of networks, at least from these heavily statistical approaches. So now the computational requirements are a major bottleneck. And to finish, I want to acknowledge my students and my collaborators, James Dignan, who's at University of New Mexico now, Jianrong Dong and Kevin Liu were my former postdocs, Matt Han at Indiana University. We did the analysis of the mosquito data set, and with Heidi Mute in New Zealand, we did the work on the biallelic markers and analyzed her data set of Oresia species. And highlighted in red here are two of my former PhD students, Dingsha Wen, Yun Yu, and a current PhD student, Jeffan Zhu. These 3 are really the engine behind all this work. They did all the work on the likelihood and the Bayesian inference from sequences, from biallelic markers, and all of that. And of course, I want to acknowledge the funding agency, especially NSF, and the Guggenheim and the Sloan Foundations for supporting my work. And with that, I thank you for your listening.
Right on. Thank you very much, Luay. That was a very clear talk. I really liked how you motivated it through the gene tree, species tree work. So
thank you.
We're taking questions now, which you can post either on Twitter or on the IRC channel as described on the Phylo Seminar website. Maybe I'll just start things off, and I might have missed this, but what is known about identifiability of these models?
So we touched on the notion of identifiability in the very first paper that we published in PLOS Genetics in 2012, that for example, in that paper with James Dignan and my students, where we looked at the probability distribution of gene tree topologies and networks, you can run easily in unidentifiability of the network and its parameters, for example, for 3 taxa and 4 taxa, because with 3 taxa, you have 3 gene tree topologies, and basically you have very little information to infer the topology of the network, every branch length in it, and the inheritance probabilities. More generally, for the identifiability of parameters, how much data you need, and so on, we have not done any work on that. In all these simulations that we run, we look at the number of reticulations we estimate, the topologies we estimate, and so on. The methods seem to be performing well. I would expect that, beyond 3 or 4 taxa, I think you would have enough information, at least from a mathematical perspective, from the loci to infer the parameters of these networks. Now, one major question here, which I pose to the mathematicians who are interested in this, the notion of statistical consistency here can be, for me, it's not even well defined, in the sense that for the case of trees, we think about statistical consistency as the amount of data increases, what's the quality of the tree we get, with what probability we get the true tree. Here, in the case of networks, as the amount of data increases, there is one parameter that could affect how we define statistical consistency, which is the number of reticulations in the network. So for example, if I look at it from maximum likelihood perspective, if you keep adding reticulations beyond the true one, suppose you got the true network and you keep adding reticulations, you cannot make the likelihood worse. So I don't know how the notion of statistical consistency, as you add more data, you can still add more reticulations also to improve the fit to the data, but now you're getting farther from the true phylogeny. But these are all interesting questions that I think they are still all open in my opinion, and will be worth pursuing and investigating.
Yeah, that's a good point. Right. I mean, I guess, yeah. I mean, for the theory, you'd need to have some sort of penalty. whether it's like a regularization penalty or some error or something like that.
Yeah. Yeah, again, in our likelihood, the work in the original one, we did, AIC, BIC, this kind of information criteria. But I would say that I haven't seen any evidence that these really work in practice for this problem.
Right. Yeah. So were the mosquito biologists convinced by your revised version of their data?
Yeah, so when we did the work with Matt Hahn, for example, when he saw the reticulation that we got it in the opposite direction, then he looked back at the data and he said, yeah, this makes sense, which illustrated the complexity of these problems that, even on 4 taxa and 5 taxa, teasing apart just by visual inspection, teasing apart incomplete lineage sorting from patterns of hybridization is impossible. Because this is a question of distributions of gene trees. This is not a question of how the gene trees differ, right? So I think that, again, in the conversations with Matt, who was a co-author on the study itself, yeah, he became convinced after the method gave this hybridization that, yes, in the opposite direction makes more sense. One other issue that we highlight in the study itself, in the molecular ecology, is the notion where, in their original study, as I said, they still focused on the notion of a species tree. They said, we have a species tree. Let's now try to postulate hybridization on top of that.
Mm-hmm.
I did not emphasize that much in my talk or my slides, but when we infer network, the nice thing about these networks is that our methods do not operate by first searching for a species tree and then finding horizontal edges. They are truly looking for DAG structures. So they don't designate anything as species tree. So when we did that and when we inferred the network basically showed that if you stick with their species tree, then the inheritance probability on the horizontal edge is about 98%. So what this basically says, that 2% of the genetic material is coming down the species tree and 98% is coming down the horizontal edge. To me, that opens up an interesting philosophical question, which is, why is that still called the species tree if only 2% of the genomic material is evolving down it, whereas we don't take the horizontal edge and call it part of the species tree?
Mm.
So that we left it, not left it open, but we put that as a discussion in the paper because I see networks as one of the nice things about networks is that you don't have to designate anything as species tree. It can infer these networks. Of course, there's great work recently by, for example, Mike Steele and others about which networks are really trees plus horizontal edges and so on. That's mainly mathematical work, but that's, I think, is a very interesting question also for the biologist.
Right. One question I had is, I noticed if I understood your prior correctly, it was like the inverse of the number of reticulate networks with that number of leaves and reticulations. And then there was another parameter on top. Is that, like, is it obvious why you would want to do that? It seems sensible.
This one?
Is it obvious why you would want to take the prior of that form? Yes.
The number of reticulations?
Yes.
So the Poisson part is that because we really wanted to regularize this thing, to penalize the complexity. Why we are normalizing by the number of networks with N taxa and M reticulations? Yes, we needed that because to account for the fact that when you go to networks with M plus 1 reticulations, the space there is much larger. So the number of networks with M plus 1 reticulations was much larger. And let me also, again, even though I did not cite every piece of relevant work, but everything here we really built on great work in the literature. This is analogous to a great paper that Mark Holder and Paul Lewis and others did on a Bayesian approach for sampling the posterior of binary and non-binary phylogenetic trees. So when you are sampling the space and you allow for polytomies in the trees, then the similar thing happens, right? I mean, the number of parameters shrinks and expands, right? I mean, you can look at a star tree, you can look at a tree with 2 branches, and so on. And in their work, in a similar fashion also, they had to normalize by the number of trees with a given number of internal branches as well.
Right.
And when we tried it, by the way, without the normalization, it does not work.
Mm-hmm. Yeah, that makes perfect sense. So what's next? I mean, I feel like you've sort of, replayed the history of phylogenetics in 10 years, like starting with maximum parsimony approaches, going through likelihood, and then Bayesian, and then quite fancy Bayesian. What are your plans after this?
Yeah, I mean, this is exactly what I have been trying to do for 10 years. My vision was, I said, I want to somehow to try to put phylogenetic networks in terms of the computational inference methods really on par with methods for trees. The community, of course, I cannot match what the community did in trees, but I wanted to elevate the work on networks to that level. Moving forward, the biggest issue here is the computational complexity. These methods, we cannot now do Bayesian inference or maximum likelihood inference of networks on, say, 15 taxa even, right? And biological data sets do have that size. In fact, there are biological data sets today with 200 and 300 taxa where biologists are asking about hybridization. This is not something we can run these methods on. Pseudo likelihood can run on these sizes, but of course the space itself is very complex. So for me, there are 2 issues here, 2 major directions to look at. One of them is the computational aspect. How do we tame the complexity of these and allow inference, large-scale inference? And of course, this comes with all sorts of, I would call them secondary questions here, including real prior of similar to birth, death, and so on networks, and all sorts of these kind of questions. But another big direction, which I think is what, for example, Cécile Année is going to talk about in the 3rd talk in this series on networks, is that now that we are building these networks, how do we use them? So I believe Cécile is going to be talking about some work on trait evolution down these networks. And I think that's a great direction to take networks to as well.
Cool. Yeah, I mean, one other thing that occurs to me that you can do with this setup that it's not interesting to do with trees is the figure that you showed with mosquitoes. The biologists had demarcated which parts they thought came from what genomes and which parts had been introgressed. I mean, that's a really hard problem as well, as if you need a harder problem, but that's also interesting.
Yeah, so we did that actually in the molecular ecology paper. I did not show the figures here. So in that paper, we show panels where for every locus along the genome, we show the path it took through that network, right? Because down a tree, the path from the leaves to the root is unique. On a network, the path from the leaves to the root, you can have an exponential number of paths.
Right.
And yeah, so in that paper, actually, we did that. We did it in a statistical approach. We said, which one of the coalescent histories has the highest probability? And we report that as the path that it took. You can think about it as also in a Bayesian way and try to give a posterior distribution of these different paths and so on. But yeah, I think that's also a very interesting question, especially for biologists looking at the notion of adaptive hybridization, which genes that were introgressed and led to some adaptive advantage, for example. Because that would be the first step is to try to find within the genomes which loci really were introgressed. And again, the picture I'm showing here on this slide is very simple, but imagine this is a network with 10 reticulations. You can imagine the number of paths become just impossible to just look at it and visually say it could have come this way or that way.
Yeah. I mean, like you say, lots of things to do and lots of work to do to make things fast. Yeah. Well, thank you very much. I think we'll end there. The next talk is Wednesday, November 22nd from Celine Squinovaca. So hopefully we'll see some of the same folks there. All right.
Thank you so much.
Thanks, Luay.
Mhm.